Good evening, and welcome to a Virtual Marine Discoveries series lecture. My name is Stefan Sewell. I'm the Community Outreach and Education Program Manager here at the National Marine Science Center with Southern Cross University. Before we get started with our evening's presentation, I'd just like to acknowledge the local custodians of this land, the Gumbaynggirr people, and pay my respects to those past, present, and future elders. Tonight's presentation is an interesting one. We have two of our PhD candidates here at the National Marine Science Center giving a presentation on their research and findings of water quality of our Coffs Coast creeks. They were able to look at 11 creeks in our immediate area and understand the impacts due to agriculture and the runoff and the consequences of that runoff. So please sit back, enjoy tonight's evening presentation.
Tonight we're going to discuss water quality and nutrient sources in Coffs Harbour coastal creeks. So I'm Shane White and my associate here is Prakton Wadnica, and I just want to acknowledge all of the co-authors on both of these reports. There's a lot of work that has gone into this, and we were funded by the Environmental Levy Grant from Coffs Harbour City Council. We're part of the Barefoot Biogeochemistry research team at Southern Cross University, and for part of this research we reached out to our colleagues at Monash University to help us with the fingerprinting techniques that we'll talk about later on. All right, so let's jump into nitrogen and phosphorus. So nitrogen and phosphorus can come from many sources, and they can run off into our estuaries. And so the diffuse sources that can come are from waste discharges, from atmospheric emissions, from agricultural inputs, and from urban development. And when we're talking about agriculture in general, 100% fertilizer uptake in any crop is not achievable. And there's generally about 20 to 60% fertilizer loss to the atmosphere, to groundwater, or to creeks. And so this is a slide from the Queensland Government sugarcane support, and shows even with these three different irrigation techniques, so we've got furrow irrigation, overhead irrigation, and drip irrigation, there's still the propensity for nutrients to run off into streams via overland flow and then end up into our marine environment, but also into our groundwater and slowly move out to our marine environment. So I'll give you a little bit of a background. We've been working on this for quite a while now. In 2017, we did a study on Bucka Bucka Creek, just northwest of Coffs Harbour. And we found that nitrate concentrations in the wet period here were more than 100 times the Australia and New Zealand Environment and Conservation Council trigger values. So these trigger values show us anything above the trigger values can lead to ecosystem degradation. So they're a guideline value that we look at to make sure that our ecosystem is in check. In 2018, we did a study in Double Crossing Creek, which is just upstream of Hearnes Lake. And we found 55% of the nitrate samples downstream of farms were at least 50 times higher than the ANZAC trigger values. So we asked nitrogen and phosphorus. These are good things. We use them as fertilizers, but they can produce some bad effects as well. And particularly in enclosed waterways with low flushing, they're particularly susceptible to algae blooms. So algae is just like any other plant. It requires nitrogen, phosphorus, and carbon to survive. And when we give them the right environment, a low flushing environment with all the food sources they need, we can have algae blooms such as here. Now this isn't currently happening, but the propensity for it to happen is there. And when this does happen, we can have low oxygen and fish kills. And so this happens when you have a large body of algae and the algae starts to slowly die. The little bugs in the soil start to eat up the algae, and as they eat up the algae, they use up the oxygen in the water, and it can lead to not enough oxygen in the water column, and so fish can't breathe, and that's when we have our fish kills. So algae blooms, or eutrophic on this map, so this is in the orange circles, and low oxygen or hypoxic in the red circles. areas are common near fertilizer-intensive land uses across the world. It's not something that's happening in Australia, it's happening all over the world. And Australia is no different. We have fertilizer-intensive land uses, and we have these similar problems. We can see in North America and in Europe and in Asia, there's a lot of these sort of issues, but they're the population centers and they're where we grow a lot of our food in the world. So these fertilizer-intensive land uses can have bad effects on our waterways. The other thing that can happen is we can have blue-green algae or cyanobacterial blooms. So this happened in the Swan River in Western Australia, and the authorities there had to put up a health warning saying this water contains harmful algae, do not fish, do not collect crabs or shellfish from these waters. It can also be harmful to humans to swim in these waters. So this is a paper from 2019 showing the distribution of cyanobacterial blooms in freshwater areas in Australia. So it is common and it does happen in these areas. So today we're going to be using these three symbols down the bottom, and they're based on the guidelines for managing risks in recreational water, and they look at the water quality in general. So you've got very good, good, fair, poor, very, very poor, and they relate to green or surveillance mode, amber or alert mode, and red, action mode. So in surveillance mode, everything's okay, we're just watching what's happening. In amber mode, we've got to start thinking about what we can do. And in red, action mode, we need to put some rehabilitation in place, otherwise we're going to have ecosystem degradation. So we'll look back at the Hearnes Lake catchment report just quickly from 2018. So on the right-hand side here, we have Hearnes Lake next to the ocean. It's an intermittently open and closed lagoon. So most of the time it's closed, but it does open to the ocean during high rainfall events. And on the just left of Hearnes Lake, we've got the yellow dot there, which is the sample site we had. We set up a little shed next to the creek there, and we took water samples. And upstream of that, we see the land classifications. And we can see there's a lot of blueberry farms in red, there's horticulture hothouses in black, there's abandoned farmland in orange, there's banana farms in yellow. And so there's a lot of fertilizer intensive land use happening in this catchment. So here's the nitrate or nitrogen results that we had. And so in the gray bars, we've got the dry. In the black bars, we've got the rain events. And in the white bars, we have days after the rain, which we classified as wet. And we can see this is the Anzac guideline here. So here it's 2.85 for this creek. And all of our samples in the dry period were in our surveillance mode. They were below the Anzac guidelines. But then we had some of our samples that were up to 50 times the Anzac guidelines, which puts us in alert mode for this time period. But then we also had some of our samples that were more than 50 times the Anzac guidelines, which puts us in action mode. So really we need to do some further remediation and look at what we can do to alleviate this problem. So we've looked at a couple of little case studies, but regional studies are really important as scientists to be able to work with government and identify hotspots for rehabilitation. And that's what Practon's done, and he'll tell us about that now.
Thank you, Shane and Steve. So I will directly go into the presentation. Here is a coastal catchment conceptual model. Here we have generally environmental scientists. We classify coastal catchment into three zones. Headwater zone, where the creeks originate. Estuarine zone, where creeks meets ocean, where the water is saline. And in between, there is a transition zone. Normally in large rivers or in big creeks, 60 to 70% of water comes from headwater zone and transition zone. Interesting thing is that in Coffs Coast region, the Estuarine zone is a part of solitary island marine park. Therefore, we decided to take a water sample just before water enters into the Estuarine zone. So the overall objective of this study is to quantify catchment nutrient runoff and assess nutrient carrying capacity of 11 freshwater creeks. We did sampling from Upper Corindi Creek up to the Pine Creek in the south. We classified these catchments into three different groups, such as forest catchment, where we have most pristine catchments such as Aravara, where we in peri-urban catchment we have most urbanized catchments such as Coffs Creek, and we have agriculturally dominated catchments such as Double Crossing Creek, Pine Creek, and Bulgulka Creek. So here is our experimental sampling design. On the left-hand side, we have runoff. What the runoff means? The runoff, it tells us how much water is lost via surface runoff in rainfall. Rainfall per day is shown along the top as a bar. And along the bottom, we have the time. We did 15 surveys, which is shown here in the blue triangle. On left-hand side, we have first 3 months classified as a dry period. And on the right-hand side, we have next 2 months classified as a wet period. So here are my nutrient results. Here we are showing average surface nutrient concentration in Coffs Coast creeks during dry and wet period. Here on the left-hand side, we have 11 freshwater creeks from Upper Corindi Creek in the north and Pine Creek in the south. The samples that we collected in a dry period, those are shown in the black color, whereas samples that we collected in a wet period, that is shown in a gray color. At the bottom, we have different nutrient concentrations, such as DIN, that is dissolved in organic nitrogen. It is nothing but nitrate and ammonium. This is very common in an environment, but it might be sourced from agriculture or surface runoff. Here, especially in this study, we found that higher dissolved in organic concentration during wet period. When we look at these all creeks in detail, then we see that in Bulgulka Creek and Double Crossing Creek, we found the nutrient concentration was dissolved in organic concentration was multiple times than any other creek. Let's move to dissolved organic nitrogen. Dissolved organic nitrogen is a form of nitrogen which is bound to the organic matter. This is commonly result of degradation of leaf or biological material. Here we found exactly opposite pattern that we found in dissolved in organic nitrogen. Here higher dissolved organic concentration that we found during the dry period. Here we have TDN, that is total dissolved nitrogen. It is composed of dissolved inorganic nitrogen and dissolved organic nitrogen. Here we have similar like dissolved inorganic nitrogen. We have higher concentration during wet period. And again, the same creek, Bulgulka Creek and Double Crossing Creek, they are behaving different than the rest of the creeks. It makes us curious. Why these two creeks are behaving different? Why the nutrient concentration or the nitrogen concentration is multiple times higher than any other creeks? Is it a groundwater? Because all over the world, many studies found that groundwater can be the source of nutrients. So is that a case here? Or is it due to the recirculated grey water? I think Shane will explain that in more detail. In the last, we have phosphate. It is a common form of phosphorus. It is present in environment normally, but it can be also sourced from agriculture and urban runoff. Here we have ANZAC water quality guidelines. That is shown here in the dotted red lines. What exactly ANZAC water quality guidelines tells us? It tells us the risk of ecosystem degradation. Overall, in this whole research period of 105 days, we found that 35% of nitrogen and 85% of phosphate samples collected during wet period are well above ANZAC water quality guidelines. So far, we have seen in surface water there is a elevated nutrient concentrations. So what are the drivers of these nutrients? Here we have black dots which represent the individual observation, while larger symbols that represent the average concentration. On the left-hand side, we have nutrients like dissolved inorganic nitrogen and phosphate, and at the bottom we have rainfall, dissolved oxygen, and radon. The radon is a groundwater tracer. It will tell us about the groundwater. Here we have very weak relationship between nitrogen and radon. What it tells? it tells us that the groundwater is not a culprit here. It is actually a good thing for us because it is also telling us that local aquifer or the shallow aquifer is not contaminated yet. When we look at rainfall and dissolved in organic nitrogen, over there we found there is a strong relationship. Again, what it tells? It tells us that nutrient in these catchments are likely driven by rainfall. I think you remember in the previous slides, we have Double Crossing Creek and Gulgulga Creek. They were showing elevated nutrient concentration. So what is the common in between these two creeks is the land use. So here we are looking, is land use is the driver of that nutrient enrichment? Here in this plot, the average of Forest Creek appears as a white circle, peri-urban catchment appears as a gray triangle, and Agriculture Creek appears as a black square. Here we found that the dissolved organic nitrogen increases with the agricultural land use. Therefore, we can say that with more agriculture, we see more nitrogen in the creeks. Let's look at the catchment exports of nitrogen. Here we have three groups of catchments: forest, peri-urban, and agriculture. When we look at the nitrogen export during the dry period, we see that the catchment export is pretty much similar with agriculture, peri-urban, and forest catchments. But when we look at the wet period, we see that the agriculture catchment export during the wet period is multiple times higher than that of the forest catchments and peri-urban catchments. That elevated agriculture catchment export during wet period, when we compare it with a global model, we can compare those with China, Europe, or India, where population density is very high. Ideally, with this model, the east coast of Australia, the catchment export should be in the range of 20 to 50. But here, we are getting catchment exports from agriculture catchments. It is in 100 to 200. That is alarming. We use sophisticated nitrogen isotope tracing to fingerprint the sources. There are possible sources of nitrogen, that it can be rainfall, it can be soil, it can be fertilizer or treated or untreated sewage. We found positive strong relationship between nitrate isotopes and percent agriculture. Again, what it tells us that agriculture activities control nitrogen in the catchments. Here we compare our nutrient concentration with ANZAC water quality guidelines. ANZAC water quality guidelines, the trigger values, it tells you the risk of ecosystem degradation. Here we have three different modes. Surveillance mode, in which less than 10% of samples are above ANZAC water quality guidelines. We have alert mode, in which 10 to 15% of the samples are above ANZAC water quality guidelines. And the most important, the action mode, in which more than 50% of samples are above ANZAC water quality guidelines. Here, for us, the most important thing is that to look at the total dissolved nitrogen, because total dissolved nitrogen is a limiting factor, especially in the estuaries and coastal ocean. What does it mean? It means that it controls the pace at which algae and aquatic plants can be produced. So when we look at the total dissolved nitrogen in detail, we found that most of the Coffs Coast creeks from agriculture and peri-urban catchments are in alert mode. And most important thing, the double crossing creek, it is shown in the red color. So it is in action mode. When we arrange these all creeks in order and by comparing them with the ANZAC water quality guidelines, we found that 53% samples of double crossing creeks are above ANZAC water quality guidelines. That needs most attention. In addition to that, the Gulgulga Creek, Coffs Creek, and its tributaries needs attention. Whereas we have more pristine creeks such as Pine Creek or Upper Corindi Creek, they are in pretty much pristine stage. So concluding all this with this, we have land use and episodic rainfall events are the major drivers of nitrogen exports. Groundwater is not a source of total nitrogen into the surface water. And it appears that the aquifers are not yet contaminated. That's actually the good sign for us. Agriculture catchment exports 3 to 6 fold times higher than that of the forest and peri-urban catchments. In overall, the nutrient enrichment seems to be an issue in most affecting streams and in peri-urban and agriculture catchment. And specifically, I wanted to mention that we need more research in a double crossing creek. Here onward, the Shane will take you to the double crossing creek, and we will go through the recommendation. Thank you.
Hi everyone, back again. Okay, so here we have Hearnes Lake. So Hearnes Lake is just downstream of Double Crossing Creek. And the most important thing to mention here is that Hearnes Lake is part of the Solitary Islands Marine Park Habitat Protection Zone. So the Habitat Protection Zone extends into Hearnes Lake. So as a community, it's really important for us to be able to protect this habitat. So this is the catchment upstream of Hearnes Lake. And we can see it's a fairly fertilizer intensive land uses. It is a mixed use catchment. So we have blueberries, we have cucumbers and tomatoes, we have bananas, and we have country that was ex-bananas. And so there's quite a lot happening in this catchment and quite a lot of fertilizer intensive land use happening here. So previously we spoke about our site in the lower catchment, but now we're going to look at the southern arm of Double Crossing Creek, and we'll just zoom into that a bit more here. Okay, so this is the southern arm of Double Crossing Creek, goes down into Hearnes Lake, and we can see here that we have, again, the hothouse horticulture, the blueberry farms, avocado farms, banana farms, and abandoned farmland. And so what we did is we treated this as a transect. So we had site 1 quite high up in the top of the catchment, then coming down to site 2, the creek branches off into 2 sections there, so we put a control site in. Then we had site 3 where we had our time series, and site 4 and site 5 in the downstream section of the creek. The time series station at site 3, we took 222 discrete samples over 91 days. And so in the dry periods, we were taking samples in the morning each day. And then when we had rain events, we'd increase our sampling rate to hourly. So we were really trying to capture that flow that was going to happen through our rain events. So we set up a temporary laboratory next to the creek. This is all our instrumentation that we use. And on the right-hand side here, this is Double Crossing Creek when we had a bit of a rain event, and we had a constant flow of fresh creek water from a pump in the creek going into our equipment. This is just a small excerpt of the data that we produced in the report. And so let's go through this a little bit. So we have our rainfall up the top here. We have our creek discharge, or the water flow in the creek. We have the soil moisture in the surrounding catchment. And we have the nitrate in the creek. So nitrate is just a form of nitrogen. So in this section, we have the first 81 days. And not a lot happened in that first 81 days. So then in the last 10 days, we zoomed in. So here's where all the action happens. So let's talk about the dry period. Here we've got this dry period, and then we had our first flush period, and then we had our second flush period. So the dry period was characterized by low creek discharge. And that was really quite interesting because we had multiple 15 to 25 mil rain events during this dry period, but there was no change in our creek discharge. So that's why we classified it as dry. And what was happening here is that we had a really high intensive rainfall event before our sampling started. And so our soil moisture was quite high in the catchment. But then for the first month or so, there was virtually no rain and it was quite hot. So our soil moisture was constantly dropping. So pretty much like a dry sponge. And then we had these rain events that happened during this period, and that only increased the soil moisture. So basically the soil was acting like a sponge and just taking up that water, and it wasn't exporting it to the creek. And that's why we see no change in the discharge. But what's interesting here is that nitrate at the time series site was consistently around 50 times the ANZAC guidelines. So the ANZAC guidelines for this creek are 2.85 micromoles. So this green line down the bottom here. So because we were so far above the ANZAC guidelines, this puts us in alert mode. And because it was constantly at that rate, we considered this alert mode. So then we move on to the first flush. So the first flush occurred with a 60 mil rain event in a day. And soil moisture was high because we had all of those earlier rainfall events during our dry period. And there was a spike in creek discharge. So basically our sponge of soil had filled up, and now we had the 60 mil rainfall event, and the sponge wasn't able to hold any more rain. And what happened here was nitrate in the time series site went up to around 100 times the ANZAC guidelines. So this puts us in action mode. So again, action mode is looking to do some remediation within this catchment to stop this happening. What's interesting is that the second flush was a smaller rain event at 40 mils. But because soil moisture was already very high from the previous rain, there was a very large spike in creek discharge that was larger than the first flush. And here, creek nitrate went up to more than 150 times the ANZAC guidelines. So this again puts us in action mode. So our theory here is that during our dry period, our crops are consistently fertilized. Our soils are storing nitrate, but there's still consistent leaching to the creek. And in our first flush and second flush, the soils are at near full water holding capacity. And the nitrate storages are being flushed from the soils, much like squeezing that wet sponge. And there's high nitrate leaching coming out to the creek. So now we'll quickly look at the 14 spatial sampling campaigns we did over 71 days. So this is using our sites 1, 2, 3, 4, and 5. And again, we did weekly sampling during our dry periods. And then in our wet periods, we would go and do these samples twice a day. And for this analysis, we're treating the creek as a transect. So we're assuming that the water flows from site 1 past site 2, 3, 4, and 5. So here's a heat map of our data, and let's explain that a little bit. So down the bottom here, we have the days of the sampling campaign. So this is the first 64 days. And on the left-hand side, we have the distance from the furthest upstream site. So what that means is that this section of the heat map represents site 1. This section represents site 2. This section represents site 3. This section represents site 4. And site 5 down the bottom here. And the colors represent the nitrate load. So when there's lots of nitrate in the creek, we're getting the orange and reddy type colors. And when there's not much nitrate in the creek, we're getting our blue and purple colors. So this is the dry period. And we're assuming again that water flows from the top all the way down through the bottom. What I found most interesting here is that our samples in the dry period at site 1 were around 7,000 times the ANZAC guidelines, which puts us in high action mode. When we saw those samples, we thought, whoa, that's pretty high. But then we looked downstream and we saw in a dry period, as the water moves down through the system, the water entering Hearnes Lake is only around 5 times the NZ guidelines for nitrate. So that showed us that the creek is providing a really valuable ecosystem service. But still, because we're more than 5 times the NZ guidelines, we considered this alert mode. So the creek has a real high natural attenuation capacity. And we found that 83% of the Site 1 load is removed per kilometre of creek as the water moves downstream. And as I said, the creek is providing a great ecosystem service by attenuating this nitrogen naturally via denitrification and plant uptake. And there's low loads delivered downstream. This is just in the dry period. And then we move into our first flush period. So in the first flush, it was quite interesting that we only dropped to 81% of the Site 1 load per kilometre of creek was able to be removed. So the creek is still providing a great ecosystem service, even with the increased flow and the increased loads. In our second flush, this is where we saw the nitrate really moving downstream and the capacity of the creek to attenuate this nitrate really diminish. Here we saw around 60 times the NZ guidelines of nitrate flowing past Site 5 and down into Hearnes Lake. So this puts us in action mode here. So really what we learned from this is that the creek has a great capacity to attenuate during our dry period, but the capacity to attenuate really drops when the flow increases. And so the attenuation capacity dropped to 52% of the Site 1 load per kilometre of creek. Even though the creek attenuation capacity drops, we still had some attenuation, but we still delivered large loads of NO3 nitrate to the downstream estuary. And then we thought about this and thought, if there's such large loads being delivered downstream, how much fertilizers are the farmers losing? So we looked at the footprints of all the potential land uses. So cucumbers, bananas, blueberries, tomatoes. And then we looked at the recommended fertilizer inputs to those industries. And we added all these potential sources up to estimate the fertilizer losses to the creek. And if we remember back to the start of the presentation, fertilizer loss is generally around the world between 20 and 60%. What we found here was more than 100% fertilizer loss to the creek. So this is theoretically impossible. Either farmers are using far more fertilizer, our assumptions are very much underestimated, there's historical nitrate leach from soils, or there's another potential source, or a combination of all these things. So we did a bit more investigation, and we found out that there's high nitrate greywater, which is recycled sewage treatment plant water, being pumped up to the farms and used as irrigation source. And this is around 2,400 meters cubed per day. So to try and isolate the sources of the nitrogen in the water, in the creek water, we use sophisticated nitrogen isotope tracing to estimate the contribution of these sources. And each of these sources has a distinct fingerprint that we can see. So we worked out treated sewage or greywater or fertilizer. Those were the two that we were looking at here. What's really interesting is that we found out that creek nitrate load was around 50% greywater and around 50% fertilizer. But as we saw earlier on, with the changes that happened in the dry and the first flush and the second flush, this didn't happen in the contribution of sources. So the contribution of sources, greywater and fertilizer, was around 50% for the dry period, the first flush, and the second flush period. It's quite interesting. So let's look at a bit of a theory on what might be happening here. And we're going to use blueberries as an example because blueberries are the dominant footprint, dominant fertilized land use in the catchment. But blueberries, they don't like nitrate. They prefer a different form of nitrogen, ammonium. So the farmers put the ammonium onto the plants, and some of the ammonium is uptaken by the plants. But soil bacteria, they also utilize ammonium, but they change it to nitrate. And nitrate is like sugar in tea. It's easily dissolved and leached out. And so some of our fertilizer nitrate can leach out of our soils and end up in our creek. But farmers here are also using greywater with, they're mixing greywater with fertilizers to water and feed their blueberries. And we sampled the greywater and found that it had very high nitrogen, and around 95% of this is nitrate. And as we said, the greywater nitrate isn't really utilized by the berries. And we have the greywater nitrate being leached out through the soils, and we find both fingerprints in the creek. And so the answer was that we found around 50% of the greywater nitrate in the creek and around 50% of the fertilizer nitrate in the creek. So the conclusions of this study show that hydrology or rainfall drives the nitrate transport in the catchment. Around 50 times the ANZAC guidelines of nitrate were found at the time series site, even in the dry period, which puts us in alert mode. And more than 150 times the ANZAC guidelines for nitrate were found in the second flush at the time series site, which definitely puts us in action mode. And the theory here is that soils are storing nitrate in the dry and then being flushed in the rainfall events. The creek is providing a valuable ecosystem service by attenuating this nitrate. But high loads of nitrate are still entering Hearnes Lake, and the probability of ecosystem impacts is very high. And so this puts us in action mode, particularly after rainfall events. And unfortunately, these are the perfect conditions for an algae bloom, which can lead to low oxygen or fish kills, or potentially a cyanobacteria bloom. So what can be done about this? There's actually a really good concerted effort happening here. We're working with state and local government to work on a lot of different recommendations, and there's a lot of on-ground works. So let's go through a little bit of that now. So we recommend to increase on-ground works to restore more than 10 meters of vegetation on either side of the creek. So studies have shown that for every meter of vegetation on either side of the creek, there's the potential to reduce nitrogen input into the creek by 4%. So if you have 10 meters of vegetation on either side of the creek, you can reduce the nitrate moving into the creek by 40%. And so this is a couple of photos of the creek just around site 1. And we can see that the riparian vegetation, or the vegetation around the creek, is in pretty poor state. There's a lot of plastic pollution, there's a lot of weeds. But funnily enough, even in this state, the creek is providing a great ecosystem service. Imagine what it could do if we restored this. As scientists, we rely on data. So we suggest increased reporting mechanisms for fertilizer use, pesticide use, and greywater use, so that we can help identify hotspots. And then we can give this to the regulatory authorities, and they can help to remediate these areas. Prakhtin and I are both working on ongoing trials of bioreactors to filter nitrogen leaving the farms. So let's explain what a bioreactor actually is. So bioreactors can remove nitrogen from the water. And in this example here, this is from the USA, and they use an ag pipe or tile drain system underneath their crops, which is on the left-hand side here. And they drain all the water from their crops and they move it down into an underground bioreactor, which is basically a big hole in the ground filled with wood chips and then covered back over with soil. And what happens here is we're creating the perfect environment for bacteria to grow. So for the bacteria, they like to eat the nitrogen and the carbon from the wood chips. And the product, in the best circumstance, is dinitrogen gas, N2, which is 80% of our atmosphere, perfectly harmless. And what happens is we get really reduced nitrate loading to our surface waters when the water comes out of these bioreactors. So we're testing this theory here. It works overseas, maybe it might work here. We've had to refine the design a little bit and do something slightly different. So here's what we've done. So this is one of the bioreactor trials that we're doing at the moment. Because in this catchment we have surface flow, and we have surface flow is the main flow to the creek. Groundwater flow has been shown to be negligible, and that's because we have such hard clay soils in this particular site. And what we did is we dug a large hole, we filled it with wood chips, we capped it with bentonite, and then we moved all the water from the farm, all the runoff water, through this structure. And in collaboration with Coffs Harbour City Council Environmental Levy Grant and North Coast Local Land Services, we've been able to do some research here, and the initial results show pretty promising outcomes. And this is the one that Practon's working on. So we've found that hothouses that grow tomatoes or cucumbers, they can be another source of nitrogen to our creeks. So Practon, again with Coffs Harbour City Council and North Coast Local Land Services, we've worked together to design a pipe bioreactor, which is basically a bioreactor that can filter and provide the right environment for bacteria to grow and reduce the nitrogen coming off these hothouses. The other thing that we found with our studies is that rainfall is really the driver. And we found that when we had the high flow events in the creek, that's when the creek wasn't able to attenuate the nitrogen as efficiently. So we suggest installing tailwater catchment ponds to reduce runoff in heavy rainfall. And how this works is basically we're trying to attenuate the flood. We're trying to flatten the curve, so to speak. And on the bottom right-hand side here, we have the flood period. And if we can slow that water down and provide a more attenuated outflow, as in the purple or pink here, we can provide the creek a better situation that it can naturally attenuate the water as it moves through the system. And how this might happen is you might have a couple of ponds that flow into each other. So the upper pond can fill up and then flow down into the lower pond, which can fill up and then slowly out to the outlet pool or out to a bioreactor or out to the creek. So these sort of engineering solutions are what we're working on currently. We're also working with farmers on ongoing trials of nitrogen use in blueberry, cucumber, and macadamia farms. And this is with the Department of Primary Industries and the Clean Coastal Catchments Project, part of the Marine Estate Management Authority's projects. And what we're looking at here is the farming inputs, so the fertilizer inputs, the soil retention, how much is stored in the soil, how much is lost to soil gases, how much is uptaken by the plants themselves, how much runs off via surface flow, and how much is lost via groundwater. And we're hoping that with this, we can provide farmers a bit more information about how much the plants actually need and give better recommendations on fertilizer use. So this is one of our little plots here. We're just doing a small experiment, which is actually quite an intensive experiment, but using this small 10 metre by 3.5 metre area to estimate the uptake and losses from this area. And finally, I really think we need to investigate the suitability of greywater use in this catchment, and particularly the high nitrate levels in the sewage treatment reuse water. Thank you. And back to Stefan.
Super. Yeah. So I'll get you guys to stay here because I'm going to stop sharing. and we'll get some, let's see. We just want to get Q&A. So you're all welcome now to, first of all, thank you for tuning in. We'll have some time now to answer some questions from you. So I see that there's a couple coming up. There's echo, okay. All right, well, let's ask any questions. So let's take a moment to see if you can, if you'd like to ask any questions, and we will endeavor to answer them. I'll give you a couple minutes there. But no, it is a fantastic amount of work that these guys have been doing. It's great that it actually is being done, and we look forward to more work with these guys to see what their outcomes can be. So any more questions? Let's see. There's a couple coming up now. So okay, great. Are any of the farmers changing from synthetic fertilizers and considering seaweed emulsion, worm tea, or mycelium?
Nicole, yeah, I see that. To be honest, I don't know, Nicole. We're working with farmers that are using synthetic fertilizers. So I'm sure that there are some that are considering these things, but no, I haven't included them in part of my studies.
Just go down. Yeah.
Yeah. Okay. So Ian Shaw's next. Is greywater being used, reticulated greywater to the Morgan's Road farms? Yes. Yeah. So that is a reticulated greywater that's instead of being output to the marine park, it's being reused on farms up here.
Okay.
Okay, Mandy Beasley, what's the reaction been from the farmers? Actually, really good. So the farmers have really opened their gates to us, and they've allowed us to do a lot of work on their farms. A lot of these guys, they want to be able to hand this land down to their kids. Many of them have been here for generations. So they're quite happy for us to come in and work on their farms and provide them with answers. It's been great. So, what mechanisms could be used to reduce the nitrate in the greywater? So there's a lot of mechanisms that could be used, and realistically, that's for the sewage treatment plants. And a lot of sewage treatment plants do use a multitude of things, including wetlands and tertiary treatments. I'm not sure exactly of the types of treatments that's used in these particular sewage treatment plants. But in speaking to Coffs Council, they're definitely looking into upgrading their systems. Yeah. What's the interest in farmers in what you're doing? So that's Jackie. Jackie, there's been a great interest from farmers, and as I said, yeah, they're very, very willing to cooperate and let us come and play on their farms. It's been really good, actually.
Next question.
There seems to be little agricultural purpose to the use of greywater on the crops being grown. John, so John, the use of the greywater is actually more for irrigation purposes. I don't think even anybody really knew the possibility of ecosystem impact of using this greywater, and I don't think it's ever really been highlighted that this greywater does have such high nitrate levels. The greywater that's leaving the sewage treatment plants is within the EPA guidelines. So there's nothing going wrong here with regard to any laws being broken or anything like that. It's all within the guidelines. So as far as I know, the whole idea was to use this water as irrigation water because we are, entering into periods of climate change and so forth, where being able to store water is becoming more difficult. When you say that an alert mode is reached during flood events, what could the response be to this alert mode in the short term? Most of the remediation you mentioned seems to be long term. So, yes, Steve, it's a really interesting area, and in the short term, I'm not too sure. We're working with a lot of government agencies to try and work out solutions, and unfortunately, this is a long-term issue. We're not going to fix it tomorrow, and so we do need to, just work through the motions and find the best way forward. What kind of treatment could the greywater go through to reduce nitrogen? So, yeah, we've kind of covered that, Nicole. So, yeah, just increasing the treatment that happens at the sewage treatment plants.
So how could that be done? Sorry.
Oh, so
yeah,
so there's a lot of different ways. Probably the best one that I can think of is the Ballina treatment plant. They're doing some really good things. They've got some reverse osmosis techniques, and they've got they run through a wetland. I think, yeah. Anyway, there is a lot of techniques that, yeah, it really is something for the sewage treatment plants to look at. But as I said, they're within their EPA guidelines. So there appears to be a need for studying the rate of conversion of ammonium fertilizers to nitrate by bacteria, as that is effectively a waste of fertilizer inputs. So John, the bacteria will change ammonium to nitrate within a couple of days. It's a natural process, and it's always going to happen. It happens all over the world. So there's really not a lot we can do about changing the ammonium fertilizer to nitrate by bacteria, because it is a natural process. And I suppose it's particularly interesting that blueberries use ammonium, because most other plants around the world, they prefer nitrate. So. Okay, is there a case for having holding ponds on the creek to regulate the water flow into Hearnes Lake in times of higher rainfall? Look, I don't know, Ian. That would be something that's probably above my pay grade a little bit, to put holding ponds in the creek itself. But yeah, definitely holding ponds for water leaving farmland is probably a more realistic option. Nicole says potentially you're going to save them heaps of money.
Yeah, with less fertilizer.
Absolutely, yeah. So the less inputs we have, the better for the farmers. That's true. So Mindy says, do you have any theories on elevated phosphorus? Was it at Arawara Creek? This one was at the forested sites.
I will
So
take that question.
take that, yeah.
Yep. The Arawara Creek is one of the pristine site that we have. It has most of the catchment is dominated by forest, but there is a lot of road works and other things are going on over there. So there is a lot of soil erosion that we can see from the satellite images as well. And in addition to that, in the soil itself in this region, we have high concentration of phosphorus. So that might be the reason that we are thinking of. That might be the reason in that case, especially with Arawara Creek.
Okay, Steve says the recommendations are really great. How can you scale up these recommendations and remediation techniques across Australia or even to other countries? So yeah, definitely that's the goal, Steve. Realistically, it's forming partnerships with government and forming partnerships with industry to be able to prove that these theories work. And once we can prove these theories work, then it's getting this into legislation that these recommendations should be used. And once we can show that this is the right way forward, then hopefully we can move on to scaling this up across Australia or to other countries. And I'm sure that we will come up with other ideas as we form new collaborations as well. John says, discharging greywater with such high nitrate levels has to raise alerts given its known environmental toxicity, whether used for irrigation or direct drainage into aquatic environments. For this site, there's also the marine park to consider. Yes, definitely. And as the council, the environmental levy grant was our funding source for these projects, we have advised the councillors. So they are now looking at possible remediation options. So our first point of call with these projects is always to advise our funding source. And so we had a meeting with council and the environmental part of the council, and they're quite aware of all these things happening. Okay. So Rose says, can we maybe get Landcare involved? There are a heap of Landcare groups on the coast, but not so many on farmland in this area. It seems to be exactly the kind of issues that Landcare was originally set up to deal with. Coffs Regional Landcare through the CHCC-EL would be the perfect way to get this happening. What do you think? Lindy. So thanks, Lindy. Realistically, I would love for Landcare to get involved. We do speak to the Landcare groups, and they are part of our kind of conglomerate of our working groups. So definitely we'll be speaking to Landcare about this and seeing what they can do, especially the remediation options in the upper part of the catchment. That would definitely be something that Landcare could get involved in. So planting on either side of the creek and so forth. So those guys are great with talking to landowners and helping with creek health. We've been involved in a few projects with them already. So.
All right. Well, thanks to everyone. I just want to thank Shane and Bracken for taking the time to come and talk to us. Thanks for your patience as well with this technology. But we'll see you in 2 months' time. We'll have our next presenter, and we'll have technology sorted out. But thanks again, guys, and we'll see you later. Cheers. Thanks. Bye-bye.